Good afternoon, everyone. Thank you for joining us for the seminar today. Our seminar speaker is Dr. Lisa Ceglia. Dr. Lisa Ceglia is a scientist on the bone metabolism team and medical director of the Metabolic Research Unit at the HNRCA. She is also assistant professor of medicine at Tufts University School of Medicine and the director of Metabolic Bone Diseases Clinic at Tufts Medical Center. Dr. Ceglia has a master's from Tufts University and obtained her medical degree from NYU School of Medicine, New York. Dr. Ceglia's research focuses on the effects of dietary and hormonal factors on the aging musculoskeletal system. She has studied the effects of vitamin D3 supplementation on human skeletal muscle morphology in older adults with low vitamin D status, as well as the effects of combined high protein and potassium bicarbonate supplementation on intermediate markers of bone and muscle metabolism. Dr. Ceglia's research work includes the impact of vitamin D3 in cardiometabolic outcomes as well. She is the recipient of several grants, including a T32 and KL2. More recently, in 2018, Dr. Ceglia was awarded an NIH funding to conduct a clinical trial on whether protein intake with or without alkali supplementation enhances muscle mass and strength in older sedentary adults. Her talk today is titled Protein Acid-Base Balance in the Diet and the Potential Effect on Skeletal Muscle in Older Adults. We very much look forward to hearing from Dr. Ceglia. Welcome.
Thank you, Sai. Okay, so I want to thank you so much for the invitation, and let me see if I can share my screen here. Okay, so it's a pleasure to be with everybody on this rainy day today on Zoom, and I want to talk about the work that I've been doing over the last decade or so here at Tufts, along with my colleagues in the Bone Lab with Bess Dawson-Hughes, and also collaborating with other labs at the HNRCA, the NEPS lab with Roger Fielding. So I have no conflicts. Let me see if I can. Okay, so starting here with this graph, I wanted to highlight the changes in skeletal muscle mass and strength with aging. And as you can actually appreciate from the graph, already starting around middle age, we see a very slow but gradual loss of skeletal muscle mass and function at rates estimated to be 1 to 2% per year for mass and about 1.5 to 5% per year for strength. And but the degree of that decline, or the slope of that change, can vary based on all sorts of factors, from genetics to lifestyle to hormone changes, and also to medical factors. And these age-related changes in muscle mass and strength have been linked to so many different factors, but I just want to focus on three main ones today. The first one being a decline in muscle anabolic hormones, specifically the IGF-1 and sex hormones like testosterone. And as you can see from this slide, IGF-1, when it binds to its receptor, it's important in recruiting kinases like PI3 kinase, and which is very important in activating, phosphorylating and activating AKT, which is an important kinase involved in regulating muscle protein synthesis and breakdown by way of indirectly activating the mTOR signaling pathway involved in synthesis, and also regulating the phosphorylation and inactivation of FOXO, which is transcription factor important in muscle protein breakdown. IGF-1 as well as testosterone are involved also in regulation of the Notch signaling pathway involved in myoblast proliferation and differentiation. And particularly testosterone is involved in regulation of myostatin, which is an important negative regulator of muscle mass. And by inhibiting myostatin, you can upregulate myoblast proliferation and differentiation. I sort of talk a little bit about this pathway because I go back to it later on in the talk at different stages. So the second important factor I want to mention is the muscle protein imbalance that we're seeing with as we age. And in this graph, you see on the y-axis muscle protein synthesis, which is MPS, and muscle protein breakdown is MPB. And studies showing that if you stimulate muscle protein synthesis using, for example, resistance exercise in a young person, shown in the thinner solid line, you can see muscle protein synthesis throughout the course here with each stimulus, and muscle protein breakdown in the dashed line. What you can appreciate is with in an older individual, there is a blunting effect of that muscle protein synthesis, whereas breakdown, in fact, is relatively stable. So this has been coined anabolic resistance that develops with aging, which is thought to contribute so much to that decline as we get older. And the third and last, but and least talked about, I think in the medical literature, is a progressive low-grade metabolic acidosis that develops as you get older, and it's driven in part by declines in renal function and also by diet. On the right here are two graphs in aging adults. You see individuals from age 20 out to age 80. And on the y-axis, these scatter plots show plasma bicarb levels over that time frame and blood pH. And what you can see is that there's an increase in endogenous acid and a decrease in plasma bicarbonate. And this ultimately results in an increase in muscle protein breakdown and a decrease in muscle protein synthesis in order to release glutamine. Glutamine is then recruited by the kidney to produce ammonia to pick up protons to mitigate the acidosis. And this is actually going back to that IGF-1 signaling pathway. As you see here, metabolic acidosis will impact this typical signaling pathway by downregulating in these solid bold arrows. You see a downregulation downstream of the IGF-1 receptor, where you see downregulation of PI3 kinase, AKT, and mTOR, resulting in reduced protein synthesis. And by decreasing AKT, you're essentially allowing the FOXO pathway to be upregulated, and that will upregulate the expression of important ubiquitin ligases that lead to muscle protein degradation in muscle cells. So the question we've posed is, can we alter certain components in the diet to mitigate these three particular factors as you get older to improve muscle health? And how about we just start with a little more protein? So as we mentioned, protein is a major building block of muscle. It breaks down to amino acids, and essentially muscle is composed of collagen, right, a protein. So that sounds reasonable as an approach. It also turns out that giving more protein results in greater circulating levels of IGF-1. This is an older study in rats where fed different degrees of daily protein intake, and you can see the rise in circulating IGF-1 levels. And of course, that would translate to a stimulation of the mTOR pathway, involved in muscle protein synthesis. And studies like this one in rats have been confirmed in human studies as well. The other aspect is by just giving more protein, it might counteract the anabolic resistance in older adults. These are studies from Dr. Phillips' lab in Canada, where you see, again, this measurement of muscle protein synthesis, where individuals young in the solid line and older in the dashed line were fed different degrees of protein. And what they measured was the change from baseline in muscle protein synthesis. And what you can appreciate is in the younger individual, you need less protein dose to reach a certain degree of muscle protein synthesis than in an older individual, suggesting that more may be better in an older person. Along those same lines, you would ask, okay, if older is better, how much more should we be giving older adults? This is a positive study out of the United States. This is the Health ABC cohort. This study was led by Denise Houston and colleagues, and they looked at 3-year changes in adjusted appendicular lean mass obtained by DEXA. And they divided the group by quintiles of energy adjusted total protein intake. So here we have the RDA group, and these are 3-year changes, but they're in fact losses, because as you can see, here's 0, here's negative. So essentially everyone is losing appendicular lean mass over 3 years. The difference is that, in fact, there's about a 40% less loss in appendicular lean mass with protein intakes of 1.1 grams per kilo per day compared to the lowest quintile at less than 0.7, well below the RDA. And this group also published about 10 years later, looking at those at the higher intakes of 1 gram per kilo per day or greater had lower incident mobility limitations over 6 years compared to those at lower intakes. So these kind of observational data are very encouraging, that potentially intakes of more than one gram per kilo per day are beneficial, along with the data showing the higher IGF-1, along with the data showing the higher muscle protein synthesis. So all of this looks very positive. However, there's a big however. When you look at meta-analyses of randomized controlled trials testing higher intakes of protein, the results are null, especially in older adults without an added resistance exercise intervention. So this is discouraging. These are 2 more recent meta-analyses where Thielen et al. Combined or pooled 8 RCTs and showed no positive effects of protein or amino acid supplementation on lean body mass or muscle strength in predominantly healthy older people. And the same for Tenhoff et al., who combined 36 RCTs and also found nothing in terms of changes, benefits in lean body mass. They also looked at thigh muscle cross-sectional area and other muscle outcomes in community older adults who are not frail. So why are so many of the intervention trials null? And I think when you're looking at intervention trials, you want to look at who the population is that's being studied. So potentially one aspect here is that we're dealing with a very healthy population of healthy aging already and very fit at baseline. And when you're studying of populations like this, you might be seeing ceiling effects with measurements of muscle strength and physical performance. Along the same lines, you might be seeing a population that's already adequately healthy in terms of their nutrition. They have adequate protein already above the RDA. And so by adding more, you're not really achieving what you're looking for as both a comparison as a placebo, and also you've already reached a certain threshold, and therefore there's not much more to gain. Of course, there's always the issue of differing studying designs in terms of how the protein, which protein was selected, how it was administered, how much was administered. These proteins, there could be animal versus plant arguments, whey versus a selective amino acid composition, and other digestibility issues. And in fact, if you look at the composition of these these foods that are being used, they have other macronutrient and micronutrient compositions that might be playing a role on impacting the muscle differently. So pooling all these studies or even trying to compare them is difficult. You're really comparing apples and oranges sometimes. Other issues might be in relation to energy balance. How much are we adding to the diet? Are we subtracting something else? And finally, I think dietary adherence is always an issue with nutrition studies. And I think diet fatigue in a longer term trial is an issue, along with altering your self-selected diet. If you're incorporating a high protein intervention, you can imagine, I think many of you in this audience know about the satiating effects of protein. And so you can imagine that maybe certain other foods might be subtracted, including protein, from diets as a result. So these are all potential reasons why we're not seeing an effect. But I want to bring up another issue here, which has we've been considering. So what if just increasing protein alone is just not the right approach here, because there might be harms in giving too much protein to an older adult population. And by harms, I'm really referring to harms to the muscle. And where I want to go with this is that the metabolism of dietary proteins from animal sources and cereal grains and seeds is metabolized to sulfur-containing amino acids, sulfuric acids and phytic acids, respectively, that therefore increase, if anything, physiological acidity. Going back to that third factor that I mentioned, this endogenous acidity that develops with aging. So if we look at different foods in this study by Schell and Anderson, I like this image that shows the potential renal acid load of different foods. And what that means is what you measure in terms of acidity in the urine excretion is an important marker from these different foods of breakdown. So as you can see, the highest potential or PROW is coming from different animal proteins and grains, whereas fruits and vegetables are more base producing. So if you combine these three things, so if you look at high protein in the diet, an aging population, and you combine that with just age-related declines in kidney function, what do you get? You get potentially a high endogenous acid production, which can actually work against the potential benefits of muscle from this change in diet. So in fact, what you're getting is some aggravation of muscle breakdown pathways from the endogenous acid production. So this is sort of just a picture of what I'm trying to describe here, that currently in our Western diet, we see a high intake of more grains and protein, and actually a low intake of fruits and vegetables, which are essentially metabolized to alkaline residues, mainly potassium bicarbonate. So I guess the next question is, well, if I'm arguing that, having more alkali in the diet could be beneficial, you might want to look at, does a diet high in fruit and vegetable intake actually impact muscle health in older adults? And over 10 years ago, one of the first papers I worked on with Bess Dawson-Hughes and Susan Harris was a secondary analysis of the Stop It trial data. This was a 3-year study, randomized controlled trial in older adults, 389 older adults who were randomly assigned to calcium and vitamin D versus placebo to look at changes in bone mass and fracture. And these individuals had 24-hour urine samples at baseline, and we looked at potassium excretion. Here's different the quartiles of potassium excretion as a surrogate of fruit and vegetable intake. And on the Y is the percent of lean body mass at baseline. And what you can see is in the highest quartile of potassium excretion, presumably the highest quartile of fruit and vegetable intake, we see the highest percent lean body mass. In a study out of the UK, the Twins UK study, over 2,500 women, this is a wide age range, 18 to 79. These investigators looked at quartiles of fruit and vegetable to protein and cereal grains. And in these women, the highest quartile of fruit and vegetable intake had the highest percent fat-free mass, or lean mass, essentially, by DEXA, supporting that concept that higher fruit and vegetable intake may be beneficial for muscle mass. But since we know that fruit and vegetables break down into these alkaline residues, why not just add alkali to the diet as a way to improve muscle outcomes? Well, one of probably the first study to look at this was a study out of UCSF by Dr. Frassetto and colleagues, where about 14 postmenopausal women were enrolled to go on to a controlled feeding study where their daily intake of protein was about 1.5 grams per kilo per day. And as you can see here, their net acid excretion was quite high at the start of their diet here, about 80 milliequivalents per day of net acid excretion. They were then all provided with a potassium bicarbonate treatment, about 60 to 120 millimoles per day. And you can quickly see the neutralization of that net acid excretion down to basically zero. And then the discontinuation of the bicarb treatment and essentially a recovery of the net acid back to baseline. And what they found was that if you look at change in nitrogen excretion, and this is a controlled feeding study, so they had a controlled nitrogen intake. If you look at their 24-hour urine nitrogen excretion, what you saw was that the change was in the negative direction here. On treatment, you see a net negative change in net nitrogen, along with a recovery, particularly the component of nitrogen that's ammonia, coming from ammonia. So not the urea nitrogen as shown in this. And this was a very clean, controlled metabolic study. And what you also can appreciate is the very rapid changes that sort of match what's going on with net acid excretion in these individuals in this controlled study. So the question came out of this study is that are we sparing muscle nitrogen as a result of this treatment? So here in our own lab with the mentorship of Bess Dawson-Hughes, we conducted 2 large bicarb studies. Actually, the more recent trial was a dose finding study where we were looking for high versus low amounts of bicarb versus placebo over 3 months. And we enrolled up to 233 adults, 60 and over. And these were adults not in a controlled setting, so they were on self-selected diets, and we measured 24-hour urinary nitrogen. We then adjusted this urinary nitrogen excretion for nitrogen intake, and we looked at the change over 3 months by dose and versus placebo. And you can see that, again, those that received the bicarb had either no change in nitrogen or a net negative or nitrogen sparing effect of the bicarb versus the placebo. In an earlier trial where we looked at a lower dose, 60 millimoles of bicarb versus no bicarb, in 162 adults 50 and over, also on self-selected diets, there was also an additional focus on seeing if bicarb impacted muscle powers in the lower extremities using a double leg press. So in this study, we over the 3 months, in the women, but not in the men, there was an interesting increase in double leg press power in these women over 3 months. Interesting, there was questions about the dose being used and whether it was adequate for the men who were on average higher weight, which led to the study on the left here, testing different doses, in fact. So based on this work in the lab and also in the literature, we hypothesized that if you combine a high protein diet along with alkali, that might just be the sweet balance between the high protein potentially stimulating muscle protein synthesis and the alkali reducing the muscle catabolism and creating that balance. And that led to a study that I worked on along with Best Dawson Hughes back now about 10 years ago. And this was a randomized controlled study. It was a crossover study, and it was in a small sample size. I will show you in a moment. And it was a controlled feeding. So all subjects crossed from either a low to a high or vice versa protein intake. And these are the amounts as 0.5 grams per kilo per day versus 1.5 grams per kilo per day. And you see in the figure here that the flipping of going from low to high and high to low. These individuals were from the outset randomized to either a potassium bicarbonate 90 millimoles or a placebo, and had a 2-week run-in period as shown in the figure, a 10-day course of one protein diet, a 5-day washout followed by another 10 days of the alternate protein. These were healthy 50-year-old and over adults. And the baseline characteristics, I mean, this was, as I mentioned, a small sample of individuals. We had mostly women in the study, an average age of 62 with, these were non-obese individuals. And this was not a weight loss study. In fact, weight remained stable and physical activity was maintained at a steady rate throughout the study. These individuals' intake, this is by grams per day, but if you divide it by their weight, on average, these individuals were either at the RDA or just slightly above, but at 1 gram per kilo per day. So not really protein deficient in this case. So what were the results of the study? And I want to first just highlight what happened with dietary protein alone in the placebo group. And we confirmed many of the changes that we would expect. So for example, starting with net acid excretion. So this is the measure of acidity in the urine over a 24-hour period. And what you see is with protein, moving from a low to a high protein, you see the increase in dietary acid load coming from the protein. You also see the increase in nitrogen excretion, as you would expect, because these individuals are actually eating more nitrogen. And then an actual significant increase in IGF-1 level, which also complements the literature showing that IGF-1 is stimulated with high protein diet. And the protein used in this study, I should mention, was actually an meat-based protein, so it was animal protein. This is the evidence that the potassium bicarb actually worked in terms of lowering net acid excretion. So in the green are those that actually receive potassium or were randomized to potassium bicarbonate, and in the white bars are those on placebo. And what you can see is that we actually didn't neutralize these individuals, we alkalinized these individuals. As you can see, the net acid excretion was lower than 0 in these groups, both on the high and on the low protein. Although, as you would expect, in the high protein group, those that were probably more acidogenic at baseline in terms of their intake, right, had a lower decline in their net acid excretion. And the main finding of this crossover study was that we found that adding the alkali to the high protein actually reduced by almost 50% that rise in urinary nitrogen excretion that accompanies the higher protein intake. And so that was very interesting. And we also saw that there was an effect on IGF-1 on both diets, just from the alkali alone. So we found that both in the low and the high protein, there was you can see here in the green bars, a higher IGF-1 level. You can see in the white bars, I had shown you these data before, that the placebo group alone, just from protein, had a rise in IGF-1. So what did we conclude from this study? So in an adult population, healthy adult population, 50 and over, on a fixed energy intake where basically we had controlled feeding, stable weight, and stable physical activity, increasing protein resulted in what would be expected as a higher IGF-1 level, a higher urinary nitrogen, and a higher net acid excretion. We also found that the bicarb reduced net acid as expected. The major findings were really that adding the alkali to the higher protein actually reduced nitrogen excretion with accompanying that higher protein load. So that might translate to a possible decrease in muscle protein breakdown. And lastly, that adding bicarb also increased the IGF-1 levels further, that might suggest that there's an effect of this combination on muscle protein synthesis as well, which hasn't been well studied. So going back to our 3 factors that contribute to muscle decline with aging, we could be mitigating these 3 factors with this combination dietary intervention, looking at the IGF-1, the anabolic resistance, and the rising endogenous acid. So that led to sort of the next step in our investigation, and that was to apply for a larger study and a longer study to really examine the effects on beyond these intermediate biomarkers, and look more at changes in muscle mass, strength and power, and performance in older adults. And the interventions that we proposed in this study was really to use a different type of protein intervention, given the larger sample and the longer involvement. To use meat-based was somewhat controversial and difficult and potentially expensive. Therefore, we used a whey protein, which is rich in leucine amino acid, which is an important amino acid in stimulating the mTOR pathway. So the premise was to add whey protein isolate at 1.5 grams per kilo per day to an individual. So it was weight-based. And secondly, was to, on the other hand, to then look at this intervention with and without alkali therapy. So we chose a dose of 80 millimoles per day as being a dose we used in a previous trial and was well tolerated by the participants. So the study schema is shown here, and it's simple. It's a 2x2 factorial. So we actually have a placebo group looking without any intervention at all here. And we have we're able to look at the independent effects of either whey alone or bicarb alone and the combination together to look at a potential additive effect. This is a 6-month trial, and it's actually the 6-month duration was chosen to look at longer outcomes such as muscle mass. And that's something that when acquired by using DEXA, a longer interval is potentially more beneficial to see significant change. We're also looking at an older population because we wanted to focus on individuals who were actually physically underactive and are at or below the RDA in terms of baseline intake, based on the fact that many of these null intervention trials have been in individuals who were already, as I mentioned, eating plenty of protein and also very fit at baseline. And so some of the measures that you capture are already at maximal effect. We're also enrolling individuals who don't have renal failure. So as you know, in renal disease, you have a further deterioration in acid handling. So these individuals are at high risk for metabolic acidosis. And so and on top of that, we're using a potassium bicarbonate intervention, and potassium handling is poor in chronic renal disease, and these individuals can develop life-threatening hyperkalemia. So we're excluding, so these are a generally healthy, older, underactive population. And these are the two interventions. Our placebo are, for the whey, we have a flavorless whey protein isolate versus a flavorless maltodextrin powder. And we have a microcrystalline placebo, cellulose placebo for the bicarb. The way we're administering the whey is not only by weight, but it's also with each meal. So there are data to suggest that giving the protein with meals distributed throughout the day might be a more beneficial way to administer it, in terms of, if you go back to the muscle protein synthesis graphs that I showed you earlier in the talk, you can imagine the giving an anabolic stimulus throughout the day might be more physiologic than doing a big bolus all at once. So I want to end. So that's basically our study right now is at a standstill right now, which we hope to resume as soon as our current crisis pandemic will slow down. But I want to end here by acknowledging my mentors and collaborators, Bess Dawson Hughes in the Bone Lab, who I met in 2004 as an endocrine fellow, and has been a fantastic teacher and research, both in clinic and research over the last 15 years. And Roger Fielding, who's been a fantastic collaborator, who's helped me work with a lot of these muscle complex exercise physiology measurements. And Donato Rivas, who's collaborated on several papers, and Norma Terran, who's helped with a lot of the grants and statistics. I also wanted to, of course, acknowledge my funding sources, most notably the recent Institute on Aging funding the R01. And finally, the staff currently on our project. Here are the members of and the units involved in this work. So thank you again for the invitation, and happy to open up to any questions.
Thank you, Dr. Ceglia, for that excellent talk. As a reminder, please use the Q&A function to post your questions, and we'd be happy to take them in the order that you post them. So the first question is from Dr. Xiangdong Wang. As comparing with your study design 3010, by adding potassium bicarbonate 80 millimole per day in the diet, how many servings of alkali vegetables and fruits that are rich in potassium do you recommend daily for the older adults in order to ensure optimal dietary acid-base balance?
Okay, well, that's a good question. So I think it depends what fruits and vegetables, they have different degrees of alkalinity, but I would say that on average, we're talking about a lot more than you would normally eat in a normal diet. It would be something along the lines of more than 10 servings of fruits and vegetables per day. So this is a much higher dose of alkali than you would get from just adding a serving to each meal of fruits and vegetables.
Great, thank you. The next comment is from Dr. Nirupa Matan. She says, nice talk, Lisa. I have a question about your animal protein randomized control trial. When participants were shifted from low to high protein, what macronutrient, carbohydrate or fat was decreased to compensate for the change?
Okay, so mainly we kept it was mainly body, I think it was fat that was substituted. And Helen Rasmussen, who's was the genius behind the diets, the low and the high protein diet, we talked about replacing some of the fat in terms of that difference, yes.
Next question is from Dr. Iris Bowles. For using vegetables, do you have to ensure you get your vegetables that are high in protein, like legumes and lentils?
Right, so there's a lot of literature about the differences between animal and vegetable proteins and differences, and what the data are really all over the place in terms of their data suggesting that the animal might, has greater, the animal sources of protein have greater impact on muscle protein synthesis, better digestibility. But in fact, I think that plant proteins also have significant value. The issue is with aging, there's concern that the digestibility of the plant proteins might be decreased, and incorporating a vegetable component, a vegetable protein, that might be an issue with aging. And I don't know that I've seen enough work in older adults to be able to make an educated or give an educated response on that. I think that clearly the animal sources of protein have higher higher dietary acid load. As you saw from that graph, if you look at where lentils lie, it's just definitely more on the alkaline spectrum. So when I'm talking about the Western diet, I'm really talking about diets that are rich in protein, but mainly animal proteins and grains, and also proteins from grains, like cereal grains, for example.
Right. The next question is from Dr. Alice Lichtenstein. She says, very interesting. There is a lot of interest in potassium and blood pressure. Are you monitoring changes in blood pressure?
So we are doing vital signs throughout the study, and yes, I'm been very interested in changes in other outcomes that may not be related to muscle directly, but indirectly. So as you can imagine, vascular flow would be very important to muscle contraction. So yes, we are tracking that.
I have a question for your RO1 RCT. How did you decide on the load for the alkali and the dosing?
I'm sorry, how are we? Sorry.
How did you decide on the dosing for the alkali provision?
Oh, so the dosing was based on a variety of, so in the preliminary trial, the crossover study, we used 90 millimoles per day, which was, it wasn't poorly tolerated, but there were a lot of participants, even over that 42-day period, who developed, some GI distress from the potassium bicarbonate. What you get is belching, burping, and what we needed to do was to decrease the dose. Not only that, but we also noticed a tremendous alkalinization, as you saw from the net acid excretion. And that wasn't really what we were trying to achieve. I think we were more trying to just achieve neutrality rather than alkalinizing these individuals. So the 80 millimole dose was used in Bess's larger randomized controlled trial using potassium bicarb, and was found to be better tolerated and didn't alkalinize these individuals in the same way. So we felt that that was a better dose.
Thank you. The next question is from Dr. Chalking Lai. He asks, is the effect of vegetable on muscle due to neutralizing effect on acid or other mechanism?
That's a good question. I have no, I think I saw some studies on carotenoids and their benefit potentially on muscle health indirectly. I've seen other aspects. I mean, there could be the alkali, but it could in fact be all these other micronutrients and what the breakdown products are of vegetables. So I think one aspect of both the preliminary trial that we conducted and the current clinical trial is that we're distilling one part of the question. Is alkali the answer to where protein might not be having its maximal effect? Is it just the acid-base imbalance? So we're just answering one part of the question, but we don't know all the potential benefits of vegetables and fruits, and there might be others. So we know, for example, and I'm going off on a tangent a little bit, but that prunes are potentially beneficial for bone health, but yet prunes don't have that high alkaline load that other fruits have. So in bone, there are a lot of interesting data about acid base in bone. And yet, so I think there's just more complexity when you're dealing with the actual food, the whole food.
Thank you. Dr. Shangdong Wang asked if you could explain how low-grade metabolic acidosis affects IGF-1 signaling and promotes muscle wasting in older adults.
I heard the latter part of the question.
So the low-grade metabolic acidosis, how does it affect IGF-1 signaling?
Ah,
Yeah, and how does it promote muscle wasting in older, and does it promote muscle wasting in older adults?
right. So the slide, and I can bring it up again, but I had a slide on, at least in severe acidotic states, like in advanced kidney disease, as an example. What you see is a downregulation in IGF-1 signaling. So you see downstream of the receptor, you see suppression of recruitment of PI3 kinase, dephosphorylation of AKT, then you see upregulation of FOXO, and that then results in upregulation of muscle protein breakdown and essentially a suppression of synthesis. In the low grade, of course, if you were to measure plasma bicarb levels in these aging adults, you don't detect a frank acidosis like you would in advanced chronic kidney disease. So these individuals don't walk around with a plasma bicarb level that's very low. And so this is a much milder process. However, although it's mild, it's insidious and it's chronic and it's long-standing. And you can imagine that the degree of breakdown is probably much slower, right? So I think of this as more of like a dose-dependent effect. So the worse the acidosis, the worse the impact on the muscle. And you see that in chronic kidney disease for sure. These individuals have severe muscle wasting. We're not seeing muscle wasting, but we're seeing muscle decline with aging, right? So it's a more gradual process, but it's present. So anything we could do to slow down that, or just to flatten that slope of that decline would be beneficial, and that's what we're proposing. I don't think I've seen studies where we're doing serial muscle biopsies where we're looking at these markers in just a healthy, in normal aging. I haven't seen a project like that.
I had two quick questions.
Yeah.
So I think the hypothesis on excess acid is interesting in terms of inducing all the pathways involved in muscle wasting. There is a much more robust literature on the role of afebrile low-grade inflammation on muscle atrophy with aging. Is there any relationship between acidosis and inflammatory levels?
Well, we do see I think that they're sort of they're related in some way, in that I don't know that I know how the process of acidosis is a stressor on the body, and in that it could induce inflammation in that way. But I don't know if the two are directly affecting each other, because I don't know the impact of alkali on inflammatory pathways per se.
Yeah, I was just sort of curious. And then the other thing that strikes me is that even in young individuals, if you put people, and this is sort of the classic nitrogen balance studies that were done in the 70s, if you put young individuals on a high protein diet, their urinary nitrogen excretion typically does increase, yet it doesn't appear as if they are wasting.
Right. So remember that their acid handling is much healthier, right? So in the kidney. So that's part of the hypothesis, is that in a younger individual, they can handle the acid load much better. In an older individual, the kidney can't adequately excrete the acid.
And then I guess some of that is what you'd like to find out in your study is, in fact, is there if you vary this excretion of acid load and or nitrogen, does it really impact total body protein stores, for example, or muscle?
Yes.
Yeah. Thank you. Really nice presentation.
Oh,
Dr. Shandong Wang asks, for the potential harmful effects that you mentioned with regards to a high protein intake in older adults, is it possible that it is due to the low carbohydrate diet, which may lead to low grade metabolic acidosis?
Right. So it's a good question, because I think that when you're what you're bringing up is that when you're increasing one aspect, you're decreasing another, and so you can't look at something in a vacuum, and so it's complicated. Yes, I mean potentially, but certainly a lot of carbohydrates have acidogenic properties. So I mentioned the cereal grains, and certainly especially most processed carbs based on wheat and so forth are acidogenic. They have a high potential renal acid load. So by decreasing, we're not actually, I mean, that's not it. So if you're talking about carbs, you're thinking about vegetables and fruits. So that's the issue. That's sort of the alkaline aspect of the diet.
Again, thank you very much, Dr. Ceglia, for the excellent talk and the discussions. Roger, do you have a question?
No, I just wanted to, again, I did also want to thank Lisa for a really nice talk, and wanted to mention that this is the last of our regular seminar seminars for this semester, and we'll be resuming again in January with the seminars. And the series has been very successful. We've had a lot of people join us virtually, and we look forward to seeing you all again in the new year.
Yes,
So
and happy Thanksgiving, everyone.
Yes.
Yeah, I want to echo what Roger said. Thanks for your participation this fall, and have a peaceful, happy Thanksgiving. Thanks, everyone.
Thank you. Thank you.
Lisa didn't have to sign out. She signed out.